Welcome to the Deep Dive, where we slice through complex information to give you the essential knowledge you need to be truly well-informed.
Today, we're plunging into the depths of an organ that's, well, often overlooked, but tirelessly works behind the scenes, your liver.
Yeah, imagine it as your body's ultimate gatekeeper, maybe a master chemist, definitely a multitasker, handling just an astounding amount of blood flow every single minute.
It truly is. It's strategically positioned. It receives blood from both your digestive tract and your general circulation. That makes it the very first processing center for almost everything that enters your bloodstream.
Okay, so our mission for this deep dive then is to unpack the liver's, let's say, intricate anatomy, its diverse cast of specialized cells, and its just astounding array of metabolic functions.
Right. We'll try to uncover how this powerhouse keeps your entire body running smoothly.
And to really underscore just how crucial this organ is, we'll introduce you to two individuals, Gene T and Amy B. Their contrasting struggles will hopefully illuminate what happens when the liver's vital roles go awry.
We'll revisit their stories throughout our conversation. Should help ground some of these complex concepts.
So let's explore this incredible organ and discover why it's truly essential to our biochemistry. Let's start with the liver's physical layout. What is it about its structure that lets it do all these incredible jobs?
Well, the liver, it's your largest internal organ, and it's mainly composed of two main lobes. These are further organized into, well, countless tiny functional units.
Okay.
But what's truly remarkable is its dual blood supply. About, say, 75% of the blood it receives comes via the portal vein.
Ah, that's the one carrying stuff from the gut, right?
Exactly. Nutrient-rich and sometimes, toxin-rich blood directly from your small intestine, stomach, pancreas, spleen, all that.
Okay, 75%.
And the rest, the remaining 25%. That's oxygen-rich arterial blood, supplied by the hepatic artery. Comes straight from your general circulation.
So it's like a central station getting deliveries from two really different routes. Everything absorbed from your gut, plus a fresh oxygen supply for its own demanding work.
Precisely. And these two blood supplies, they actually mix as they flow into the liver's unique blood channels. They're called sinusoids.
Sinusoids. And you said they're unique? How so?
Imagine these channels aren't like solid pipes. They're uniquely leaky. They're lined with cells that have these microscopic pores, tiny windows, essentially.
Okay.
And these pores act like a sieve. They allow small, important molecules to easily pass through and reach the main liver cells, the hepatocytes, for processing.
Ah, so it lets the good stuff through quickly?
Right. It lets the liver quickly access and filter what's coming in while kind of keeping larger, unprocessed particles, like big fat globules, from getting through too soon. This is a very clever design.
That is a fascinating design. Seems perfectly suited for its role. And beyond just filtering blood, the liver also secretes bile, doesn't it? How does that system work?
It does, yeah. After the hepatocytes process compounds, they also produce bile. This flows into tiny channels called bile canaliculi.
Canaliculi.
And these channels run in the opposite direction to the blood flow. It's quite neat. Eventually, they empty into larger bile ducts.
Those laid out of the liver.
Yep. They fuse to form the common bile duct, which ultimately releases bile into the duodenum, the first part of your small intestine, to help digest fats.
Right, digestion. And some get stored too.
Correct. Some of that bile is also stored in your gallbladder for release right after a meal when you need it most.
Okay, here's where it gets really interesting for me. You mentioned different liver cells. Not all are created equal, right? Each has a specific crucial role in this bustling organ.
You're absolutely right. While the liver has several cell types, hepatocytes are, well, they're the undisputed stars. They make up about 80% of the liver's volume.
80%. Wow.
Yeah, and they perform the vast majority of its metabolic pathways. These cells are incredible powerhouses, and their activity is tightly controlled by hormones like insulin and glucagon.
And they can regenerate.
Amazingly so. If the liver is damaged, hepatocytes can replicate rapidly to repair it and maintain a constant liver mass relative to body size. Really remarkable.
So if these critical hepatocytes are damaged, thinking about someone like Amy B., what kind of early warning signs might we see? In lab tests, maybe?
That's a great question. In cases like Amy's where there's initial injury, we typically see elevated levels of certain enzymes leaking out of damaged cells into her blood.
Like which ones?
Things like hepatic transaminases, ALT and AST are the common ones, and alkaline phosphatase. Also, you might see increased total bilirubin levels.
Bilirubin, that's related to jaundice.
Exactly. It builds up if the hepatocytes are struggling to process it or if bile flow is blocked. These are classic indicators that hepatocytes have been damaged or are, let's say, malfunctioning.
Okay, so hepatocytes are the main workers. What other players are there?
Well, besides the hepatocytes, we have those crucial endothelial cells we just mentioned. They form that leaky lining of the sinusoids, which is absolutely key for efficient exchange between blood and hepatocytes.
Got it. The sieve makers.
Right. Then there are Kupffer cells. Think of these as the liver's resident immune cells, macrophages, basically.
Macrophages, so they eat stuff.
They do. They're packed with enzymes, and they sit in the sinusoid lining, essentially vacuuming up things like bacteria that might sneak through from the gut, old, damaged red blood cells, other debris. They're a major defense line.
Like tiny bouncers keeping the bad stuff out.
Huh. Exactly. That's a good analogy. Next, we have hepatic stellate cells. Now, these are interesting. They're known for storing vitamin A.
Vitamin A storage.
Yeah. But they also play a big role in regulating connective tissue in the liver. We'll come back to them later because they could become problematic in chronic liver disease, like fibrosis.
Ah, okay. Foreshadowing. Anyone else?
One more key type, pit cells. These are basically liver-associated natural killer lymphocytes, another layer of immune defense, particularly against invaders like tumor cells and viruses.
So, quite a team effort going on in there. What does this all mean for us then? The liver isn't just a simple filter. It sounds like a dynamic processing plant, constantly monitoring, recycling, modifying, distributing.
It absolutely is. Think of it as the body's central receiving and recycling hub. Because it gets that first pass of blood from the digestive tract, it's perfectly positioned.
First dibs on everything.
Pretty much. It monitors and distributes nutrients, and crucially, it intercepts and detoxifies potentially harmful compounds absorbed from the gut before they reach the rest of the body.
And it makes stuff too, right?
Oh, yes. It synthesizes vital compounds like blood-clotting proteins, absolutely essential, and hamete for red blood cells, even the building blocks for our DNA, purines and pyrimidines. And remember, that leaky structure and the low blood pressure in the portal system, that ensures highly efficient exchange. Large molecules get removed, newly synthesized molecules get easily secreted into the blood. It's optimized for traffic flow.
That's quite the multitasking operation.
Yeah.
But beyond just processing nutrients, you said it's the body's primary defense against toxins. Let's dive into that.
Right, detoxification. The liver truly excels at inactivating and detoxifying what we call xenobiotic compounds.
Xenobiotics. Fancy word. What does it mean?
It just means foreign compounds,
Yeah.
stuff that isn't a nutrient but gets into your system. Could be natural components in food, food additives, environmental chemicals, or, very commonly, drugs.
Okay. And many of these are fat soluble.
Many are, yes, which means they'd otherwise tend to accumulate in your body's fat stores, potentially indefinitely. The liver uses a really clever two-phase detoxification process to deal with them.
2 phases. How does that work?
Okay, so in phase-out reactions, liver enzymes introduce or expose reactive chemical groups on the xenobiotic molecule. It's like adding a little handle or tag.
Just modifying it slightly.
Exactly, preparing it for the next step. Then, in phase 2 reactions, the liver attaches a larger, usually negatively charged group to that handle. Things like sulfate or glucuronic acid, which is a sugar derivative.
And why add that charged group?
That dramatically increases the compound's water solubility, makes it much easier for your kidneys to filter it out into urine or for the liver itself to excrete it into bile. It's essentially tagging these fat-soluble toxins for removal.
So it's generally protective, making harmful things easier to get rid of, but I feel like there's a but coming. Can these reactions sometimes backfire?
That's a really crucial nuance. Yes, they can. While generally beneficial, sometimes a phase I or phase 2 reaction can inadvertently convert a relatively harmless molecule into something more toxic or even a potent carcinogen. It's not the usual outcome, but it happens.
How does that work? What's involved?
A key player here is a large family of enzymes called the cytochrome P450 system, often just called CYP450s.
Ah, I've heard of those.
Yeah, they're primarily found in the liver, in the smooth endoplasmic reticulum. Think of them as your body's versatile chemical modification team. They're excellent at oxidizing substrates, often introducing oxygen into their structure, usually as part of that phase A preparation.
And this CYP450 system is incredibly important for drug metabolism, right? I remember hearing something about grapefruit juice affecting medications because of this.
An excellent classic example. One specific CYP450 enzyme, CYP3A4, is particularly significant. It metabolizes the largest number of common drugs in humans.
Okay.
So if you take two drugs that both rely on CYP3A4, they can compete for the enzyme. The one that binds tighter gets processed, while the concentration of the other drug can rise, sometimes dangerously.
And grapefruit juice.
Grapefruit juice contains compounds that are potent inhibitors of CYP3A4. So if you drink grapefruit juice while taking certain statins, common cholesterol-lowering drugs, that CYP3A4 metabolizes the enzyme gets blocked.
Meaning the statin doesn't get broken down?
Exactly. Its levels in your blood can shoot up dramatically, maybe even 15-fold in some cases. That can lead to significant muscle problems or liver toxicity. It really highlights how diet can impact drug effects.
That's a powerful example. What about when CYP450 actually creates something more toxic, not just failing to break something down?
A well-studied example is aflatoxin B1. This is a nasty compound produced by a type of mold that can grow on things like peanuts and corn, especially if stored improperly.
Okay.
Aflatoxin itself isn't the main problem, but a CYP450 enzyme in the liver metabolically activates it, turning it into a highly reactive epoxide form.
And that activated form is the dangerous part.
Yes, it can directly bind to and damage your DNA, causing specific mutations, particularly in a gene called P53, which are strongly linked to the development of liver cancer, hepatocarcinogenesis.
Wow, so the liver's own detox system accidentally creates a carcinogen. Are there other common examples of this kind of paradoxical toxicity?
Yes, a very common one involves acetaminophen, the active ingredient in Tylenol, and many other painkillers.
Acetaminophen, but that's generally safe, right?
At normal doses, yes. Most of it is safely processed through phase 2 reactions conjugation with glucuronide or sulfate and then excreted. No problem.
Right.
But a small fraction is normally metabolized by a different CYP450 enzyme called CYP2E1 into a highly reactive toxic intermediate. Let's call it NAPQI.
NAPQI. Okay, toxic intermediate.
Right. Normally, your liver has another defense, a molecule called glutathione. Glutathione quickly neutralizes this NAPQI, rendering it harmless. At therapeutic doses, your glutathione stores are easily sufficient.
But if you take too much?
Exactly. With an overdose, the main phase 2 pathways get overwhelmed, saturated. More acetaminophen gets shunted down that minor CYP2E1 pathway, producing way more toxic NAPQI than usual.
And the glutathione can't keep up.
Precisely. Glutathione stores get rapidly depleted. Once they're gone, the NAPQI is free to attack critical proteins and lipids in the hepatocytes, causing widespread cell death and potentially fatal liver failure.
That's scary, and I've heard alcohol makes this worse.
Crucially, yes, chronic alcohol consumption actually induces or increases the activity of that specific enzyme, CYP2E1.
Ah, so it makes more of the toxic NAPQI even from normal doses.
Correct. Individuals who regularly drink significant amounts of alcohol are much more susceptible to acetaminophen toxicity, even at doses that would normally be considered safe. Their boosted CYP2E1 produces far more NAPQI, overwhelming their glutathione system much faster.
Is there anything that can be done if someone overdoses?
Thankfully, yes. The main treatment is giving N-acetylcysteine, often called NAC. NAC works primarily by helping the liver replenish its glutathione stores, boosting that natural defense against NAPQI. It's most effective if given early.
Okay, it's clear the liver plays a massive role in managing what comes into the body, especially potential toxins, but it's also a master of internal fuel management, right? Energy balance.
Absolutely. You could argue that regulating blood glucose is one of its most paramount roles. Keeping your blood sugar stable is critical, especially for your brain.
How does it do that?
Through a really sophisticated dance involving several pathways. It can store glucose as glycogen when levels are high, like after meal, break down that glycogen to release glucose when levels drop, and even synthesize new glucose from other sources, like amino acids, in a process called gluconeogenesis.
And it responds to hormones.
Tightly regulated by hormones, primarily insulin and glucagon, and critically, the liver is one of only 2 tissues in your body, the other being the kidney that has the enzyme glucose 6-phosphatase.
Why is that enzyme important?
It allows the liver to release free glucose back into the bloodstream to maintain blood sugar levels for other tissues. Muscle can store glycogen, but it can only use it for itself. The liver exports glucose for everyone else.
So after a meal, say rich in carbs, what's happening specifically?
Well, the liver quickly takes up glucose and also converts other dietary sugars like galactose and fructose into glucose or intermediates that can enter glycolysis. There's a key enzyme in the liver, glucokinase, which acts like a sensor.
A glucose sensor.
Yeah, it becomes active only when glucose levels are high, like after a meal, ensuring the liver efficiently takes up and processes that incoming sugar surge, preventing your blood sugar from spiking too high. It's part of a brilliant feedback system.
Okay, carbs handled. Moving on to fats, then. Lipids. What's the liver's role there?
Just as central. The liver masterfully orchestrates fat metabolism too. During fasting, long-chain fatty acids become a major fuel source, processed by the liver. But there are also special cases, like medium-chain length fatty acids or MCTs.
MCTs, like in coconut oil. I hear about those in supplements.
Exactly. They're often used in nutritional supplements, especially for patients who have trouble absorbing regular fats. MCTs are absorbed differently.
How so?
They bypass the usual lymphatic transport system for fats and go directly into the portal vein, straight to the liver.
Yeah.
They provide a quick, easily utilized calorie source,
Yeah.
very helpful in certain clinical situations.
Interesting. What about really long fats?
Good question. The liver also has specialized compartments inside its cells called peroxisomes. These organelles are crucial for breaking down very long-chain fatty acids, ones that are too long for the mitochondria, the main fat-burning factories, to handle initially.
Peroxisomes. Okay, so they shorten them first.
Basically, yes. They partially oxidize these very long chains, and the shorter products can feed into the regularly mitochondrial beta-oxidation pathway. If this peroxisomal system fails, it causes serious problems.
And that's where something like Zellweger syndrome comes in, I assume?
Precisely. Zellweger syndrome is a tragic, rare inherited condition where individuals are born without functional peroxisome.
Oh, peroxisomes at all.
Correct. This leads to a buildup of these very long-chain fatty acids in tissues throughout the body, causing devastating effects, particularly on the brain and liver development. It really highlights how critical even these specialized metabolic niches are.
It really does. Now, how does the liver know how much fat is around? How does it sense and respond to fatty acid levels?
That's where another set of key regulators comes in, PPARs.
PPARs, what are those?
Peroxisome proliferator activated receptors. These are special proteins, nuclear receptors actually, that act like fatty acid sensors inside liver cells.
So they detect fats?
Yes, particularly one isoform called PPARA, which is abundant in the liver. When levels of fatty acids rise inside the liver cell, they actually bind to PPARA. This activates PPARA, which then turns on genes involved in fatty acid uptake, transport, and, crucially, oxidation, burning those fats.
So more fat signals the liver to ramp up fat burning. Makes sense.
It's a key regulatory mechanism, and we can actually target this pharmacologically
Oh.
with drugs called fibrates. These are used to treat high triglyceride levels in the blood. Fibrates work by binding to and activating PPARA, just like fatty acids do.
So they trick the liver into thinking there's more fat than there is?
Sort of. They enhance the signal, essentially telling the liver to really boost its fat-burning machinery, which helps lower triglyceride levels in the blood. It also affects certain lipoproteins involved in fat transport.
Fascinating. You also mentioned the liver using fat pathways for detox.
Yes. Sometimes the liver cleverly co-opts parts of its fatty acid metabolism pathways to detoxify certain xenobiotics that structurally resemble fatty acids. Things like benzoate, a food
Yeah.
preservative, or salicylate from aspirin.
How does that work?
It conjugates them, often with the amino acid glycine, using pathways similar to those for activating fatty acids. This makes them water-soluble for excretion. But linking back to aspirin, this isn't without potential risks, especially highlighted by Rey syndrome.
Right, Rice syndrome. You mentioned that connection earlier. Very serious.
It's really serious, particularly in children. It involves severe brain swelling, encephalopathy, and liver damage, often triggered by aspirin use during a viral illness like the flu or chickenpox.
What's the underlying problem in the liver?
It seems to involve profound damage to the mitochondria within the liver cells. Remember, mitochondria are the powerhouses, essential for energy production, including fatty acid oxidation. Aspirin, or perhaps its interaction with the virus, seems to disrupt mitochondrial function severely, leading to liver failure, low blood sugar, and a buildup of ammonia. It's why aspirin is generally avoided in children with viral infections now.
A stark reminder of how interconnected these pathways are. Okay, finally, we can't forget amino acids and other key synthetic roles.
Absolutely not. The liver is the central hub for amino acid metabolism. Most amino acids, after digestion, go straight to the liver via the portal vein.
What does it do with them?
It uses them for synthesizing its own proteins, releases them into circulation for other tissues, or if there's an excess, it can break them down for energy or convert them to glucose or fat.
And dealing with the waste.
Critically important. Amino acid breakdown produces ammonia. which is highly toxic, especially to the brain. The liver performs the vital urea cycle, converting that toxic ammonia into urea, a much less toxic compound that can be safely excreted by the kidneys. This is a primary detoxification function.
So failure of the urea cycle is a major problem in liver disease.
A huge problem. We'll see that with gene T. The liver is also the primary factory for synthesizing most of the proteins circulating in your blood.
Like which ones?
The most abundant one is albumin. Albumin is crucial for maintaining osmotic pressure in the blood, keeping fluid from leaking out into tissues. It also acts as a transport vehicle for many substances like hormones, fatty acids, and drugs.
And clotting factors.
Yes, almost all the essential clotting factors needed to stop bleeding are synthesized exclusively in the liver.
And if those aren't made properly, that connects directly back to gene T's symptoms, right? The swelling from low albumin, the bleeding risk from low clotting factors.
Precisely. Her symptoms are a direct consequence of the liver failing in these synthetic roles. The liver also synthesizes most blood glycoproteins, proteins with sugars attached.
Okay. Anything special about those?
Well, as these glycoproteins age in circulation, they tend to lose some of their terminal sugar units. The liver has a special receptor, the azole glycoprotein receptor, that recognizes these old glycoproteins and removes them from the blood, allowing their amino acids to be recycled. It's a quality control system.
Efficient recycling. One last pathway, you mentioned the pentose phosphate pathway.
Right, the pentose phosphate pathway, or PPP. This pathway doesn't primarily make ATP energy, but it's vital for two other things the liver desperately needs.
Which are?
First, it produces NADPH. This molecule is essential as a reducing agent, basically providing electrons for many biosynthetic reactions, making fatty acids, cholesterol, bile salts.
Okay, building blocks. And the second thing?
Antioxidant defense. NADPH is absolutely critical for maintaining the liver supply of reduced glutathione. Remember, that molecule needed to neutralize toxins like acetaminophen's byproduct.
Ah, glutathione again.
Yes, glutathione is a major antioxidant, protecting liver cells from damage by reactive oxygen species or free radicals, which are constantly generated during the liver's intense metabolic activity. The PPP provides the NADPH needed to keep glutathione in its active, protective state. Given the liver's constant exposure to toxins and high metabolic rate, this antioxidant defense is vital.
It's truly clear the liver is incredibly versatile, a real biochemical powerhouse. But as we've touched on, what happens when this master organ starts to falter? When it can't perform these multitude of functions?
Well, the impact is profound, often devastating, because, frankly, no other organ can fully compensate for widespread liver failure. The signs and symptoms of liver disease really reflect the loss of its many varied roles.
What are some key indicators doctors look for?
We often see those elevated liver enzymes in the blood ALT, AST, indicating ongoing hepatocyte injury or death. Jaundice, that yellowing of the skin and eyes, is common. It's caused by the buildup of bilirubin when the liver can't process and excrete it properly via bile.
We mentioned clotting problems.
Right. Increased clotting times, measured by tests like PTINR, result directly from the liver's reduced synthesis of those crucial clotting factors. This leads to easy bruising and potentially serious bleeding.
And the swelling.
Edema, particularly in the legs and abdomen, where it's called ascites, is very common. This happens because the failing liver isn't making enough albumin. Low albumin decreases the blood's osmotic pressure, allowing fluid to leak out of blood vessels into surrounding tissues.
And the brain effects.
Perhaps one of the most serious complications is hepatic encephalopathy. As the liver fails, especially the urea cycle, ammonia levels rise in the blood. Ammonia is toxic to the brain, leading to confusion, altered personality, lethargy, progressing even to coma in severe cases.
Let's bring Gene T's story back into focus here. Her history of alcohol abuse led to cirrhosis. What exactly is cirrhosis happening in her liver at a cellular level?
Okay, so Gene T developed alcohol-induced cirrhosis. This isn't just inflammation. It's a diffuse, progressive, and largely irreversible scarring of the liver. In this condition, those hepatic stellate cells we mentioned earlier, the vitamin A storing ones,
What happens to them?
chronic injury, like that from excessive alcohol, activates them. And when activated, they transform. They stop storing vitamin A and instead start producing massive amounts of collagen and other extracellular matrix proteins, basically scar tissue.
So they become scar-making machines.
Exactly. They lay down dense, rigid scar tissue, fibrosis, throughout the liver. The scar tissue replaces the liver's normally flexible functional tissue and disrupts its architecture. When the scarring becomes widespread and bridges between different areas, That's cirrhosis.
So it's not just damage, it's a fundamental physical change in the liver structure.
Well,
What are the consequences of all that scarring?
the consequences are really severe and drive many of the clinical problems. First, the scar tissue obstructs blood flow. Remember those leaky sinusoids? They get compressed and lose their fenestrations, those little pores. The whole liver becomes stiff,
Making it harder for blood to get through.
much harder. This dramatically increases resistance to blood flow coming from the portal vein. This backup of pressure in the portal system is called portal hypertension.
Okay, high pressure in the portal vein, what does that lead to?
Well, the blood has to go somewhere. So it starts finding alternative routes back to the heart, bypassing the high-resistance liver. It shunts through normally small, low-pressure veins, particularly those connecting the portal system to the general circulation, like veins in the esophagus and stomach.
And those smaller veins aren't built for that pressure.
Not at all. They become engorged and dilated, forming fragile, swollen vessels called varices. Esophageal varices are particularly dangerous because they can rupture suddenly, causing massive, life-threatening hemorrhage, like the severe gastrointestinal bleeding Gene T experienced.
A terrifying complication. And the brain toxicity you mentioned earlier, hepatic encephalopathy. How does cirrhosis worsen that?
Two main reasons. First, the damaged liver cells themselves have a reduced capacity to run the urea cycle and detoxify ammonia coming from the gut. Second, because of that portal hypertension and shunting, a lot of the gut-derived ammonia bypasses the liver altogether and goes straight into the general circulation.
So more ammonia gets produced or absorbed and less of it gets cleared.
Exactly. The ammonia levels in the blood rise significantly, cross the blood-brain barrier, and interfere with neurotransmitter function, leading to the neurological symptoms of hepatic encephalopathy.
And you also mentioned diabetes linked to cirrhosis.
Yes, hepatogenous diabetes. It's quite common in patients with advanced cirrhosis. The mechanisms are complex, involving reduced glucose uptake and clearance by the damaged liver, increased production of glucose, insulin resistance in peripheral tissues, and sometimes impaired insulin secretion from the pancreas. The liver's central role in glucose balance is severely disrupted.
What a cascade of problems stemming from that initial scarring. It paints a really stark contrast to Amy B's case then. She also had a liver issue, but her outcome was much better.
Indeed, Amy B's situation was quite different. She had amoebiasis, an infection caused by a parasite likely contracted from contaminated food or water. This led to localized liver abscesses, pockets of infection and inflammation within the liver tissue.
So infection, but localized.
Yes, while certainly serious and requiring treatment, it was a focal process, not the diffuse, progressive scarring seen in cirrhosis. Her case really highlights the liver's robust immune defense role. Her Kupffer cells and other immune cells would have been actively fighting the infection.
And treatable.
Absolutely. With appropriate anti-amoebic medication, the infection could be cleared and the abscesses typically heal. Crucially, because the underlying liver architecture wasn't destroyed by diffuse fibrosis, she was expected to make a full recovery without the long-term, life-altering consequences of cirrhosis the Gene T faced. Amy's story is more testament to the liver's resilience when faced with an acute, treatable insult.
It's just fascinating, really, how incredibly interconnected all these diverse functions are. You disrupt one part, say, detoxification or protein synthesis, and the ripple effects spread throughout the entire body.
It truly underscores why the liver is considered such a vital organ.
Well, we've taken quite a deep dive today into the incredible world of liver metabolism. We've uncovered its central role as the body's master chemist, its detoxifier, its fuel manager. It's just remarkable.
From disarming toxins like drugs and alcohol, to meticulously regulating your blood sugar, synthesizing almost all your blood proteins, managing fats, the sheer scope of its biochemical activity is mind-boggling.
Indeed. And as we saw vividly with the contrasting stories of Jean and Amy, understanding these intricate biochemical pathways isn't just, academic, it's absolutely fundamental to diagnosing, managing, and treating a vast array of human diseases.
Absolutely. The liver's resilience is amazing, but as Jean's case shows, it's definitely vulnerable to chronic insult. Its health is just paramount for your overall health.
Think about that. This single organ constantly working, making critical decisions about everything you consume while simultaneously powering, rebuilding, and defending your body. It's truly a marvel, maybe the unsung hero of our biochemistry.
I think that's a fair description.
Thank you so much for joining us on this deep dive into the liver. We really hope this has given you a clearer, perhaps more appreciative picture of this vital organ and maybe left you with plenty to ponder. And a warm thank you from the Last Minute Lecture team for sharing this fascinating chapter with us.